Looking at how the heart muscle contracts, we'll do a little review of some of our basics. So, contraction stimulated by the conduction system of the heart. It triggers ion flows across the cardiac muscle cell membrane. This initiates an action potential in cardiac muscle cells that leads to a physical cardiac muscle contraction using the same exact mechanisms as skeletal muscle. And then the heart will reset and return to baseline and starts again on its own thanks to the conduction system. So, as a reminder, the heart has three major types of cardiac muscle. Atrial and ventricular muscle will contract like skeletal muscle with a longer duration of contraction. There's also specialized muscle fibers, both excitatory and conductive, and these are automatic, rhythmical electrical discharges in the form of action potentials that control the rhythmic beating of the heart. When the heart pumps or contracts, it pumps as a syncytium, which means that when one heart cell excites, the action potential spreads rapidly to all cells. And ions will move through intracellular fluid along the longitudinal axes of the cardiac muscle fibers, allowing for action potentials to travel from one cardiac muscle cell to the next through intercalated discs. There are two syncytiums that are present to allow for the atria to contract prior to ventricles. The atrial has the walls of the two atria, and the ventricular has the walls of the two ventricles. So now let's look at how a cardiac action potential actually does its thing, right? Because that is a key component here, is thinking about how does a cardiac action potential work. So cardiac action potential, it's just an electrical or mechanical activity that's created by a depolarizing current. And we're going to look at these different phases, and we are going to work through describing what's happening in each phase. This is what we're going to be talking about first, which is our ventricular muscle fiber. So there's five phases total, 0 through 4, and they all happen in every single cardiac myocyte except for the SA and AV nodes. Depolarization occurs, which creates a more positive membrane. Then the action potential will plateau, as we'll see, this little plateau phase here, here, here. And then lastly, repolarization occurs and we have a more negative membrane. So at phase 0, that's technically depolarization. The cardiac cell is stimulated by the conduction system of the heart. Our voltage-gated fast sodium channels open. Sodium moves quickly inside of the cell, and this is a 1 plus positive charge that leads to a quick depolarization of the cell membrane, leading to the membrane potential becoming more positive, moving up our y-axis. In the background, it's important to know that during this time, our calcium channels our slow voltage gated calcium channels open, and calcium also starts to move into the cell, but it's moving kind of slow and steady. It's not as fast as our quick sodium channels. At phase 1, once we've reached the super positive level, we start to repolarize because our voltage gated fast sodium channels close and our voltage gated fast potassium channels open. So there's an efflux of 1 plus charge leaving the cell. But this is not as dramatic as it could be because calcium is still moving inside the cell in the background because calcium channels have not closed yet. Cells start a little initial repolarization dip, but then they're going to pause because we still have calcium influx. So that's where we get to stage 2 or phase 2, which is the plateau. And this is because we get low enough or we get less positive enough that our voltage gated fast potassium channels close, but our voltage gated slow calcium channels are still open, allowing for calcium influx, which will trigger calcium release from the sarcoplasmic reticulum and myocyte contraction via our excitation contraction coupling. And so that's what that plateau phase is allowing us to do. And then once we've been at that plateau phase for a little bit, our voltage-gated slow calcium channels will close once heart contraction has taken place. Our voltage-gated slow potassium channels will open, and we have an efflux of 1 plus charge leaving the cell, which brings us back to 4, which is our resting membrane potential. At this point, we have some high potassium permeability through our potassium channels. The average resting potential is between -80 and -90. So what does this look like from a cell perspective? Well, our action potential that we just generated is going to move across the surface of the membrane. When it reaches a T-tubule, it will hit essentially the L-type or slow calcium channel. So we can see that here. This allows for calcium influx from the extracellular matrix. Calcium influx from the extracellular matrix will bind to the ryanodine receptor, which is on the surface of the sarcoplasmic reticulum. And when the ryanodine receptor is activated, then calcium is going to be released from the sarcoplasmic reticulum into the cytoplasm where it binds specifically to troponin C, that's high yield. And then that's going to lead to a muscle contraction by causing a conformational change that allows actin and myosin to bind. After the plateau, calcium influx to the muscle fiber will stop, and calcium ions in the sarcoplasm or the cytoplasm are pumped out of muscle fibers into the sarcoplasmic reticulum as well as into the T-tubule extracellular fluid space. One pump that they use is the calcium adenosine triphosphate pump, or SERCA, sarcoendoplasmic reticulum calcium ATPase, that pumps calcium back inside. There's also a sodium calcium counterexchanger and a sodium potassium ATPase pump. If we compare and contrast muscle types, our myocytes are coupled electrically by gap junctions and intercalated discs. Our skeletal muscle cells are not coupled via intercalated discs. Cardiac muscle's action potential has a plateau because of calcium influx and potassium efflux. Skeletal muscle does not have a plateau. And cardiac muscle requires calcium influx from the extracellular fluid to induce calcium release from the sarcoplasmic reticulum, versus skeletal muscle has mechanical coupling of our L-type calcium channels with the ryanodine receptor, so it can stimulate it a bit easier. We'll stop here. When we come back, we're going to look at the cardiac cycle.